Our second SoftChalk lesson in Unit 1, Lecture 4 deals with the bacterial chromosome located in an area of the bacteria called the nucleoid. So let's look at the fundamental statement or bullet points for this particular SoftChalk lesson. The genome is a sum of an organism's genetic material. Bacteria contain a single chromosome of double-stranded helical deoxyribonucleic acid. The region of the cytoplasm where the chromosome is located and visible when viewed through an electron microscope is called the nucleoid. The bacterial chromosome is typically a physical and genetic circle and becomes supercoiled and is not surrounded by a nuclear membrane. Bacteria do not carry out mitosis or meiosis. DNA topoisomerase enzymes are used to supercoil and relax the bacterial chromosome during DNA replication and during transcription. Like eukaryotic DNA, prokaryotic DNA replicates by sequential unwinding of the two DNA parent strands and the subsequent complementary base pairing of DNA nucleotides with the DNA nucleotides of each parent strand. During DNA replication, the nitrogenous bases adenine can form hydrogen bonds with thymine, while guanine forms hydrogen bonds with cytosine. Genes located along the DNA are transcribed into RNA molecules. Messenger RNA, transfer RNA, and ribosomal RNA. Primarily, of course, most of the genes are transcribed into messenger RNA, since there's only a limited number of transfer RNA and ribosomal RNA molecules in a bacterium. Messenger RNA is then translated into proteins at the ribosomes, and in fact, transcription and translation are coupled in bacteria. During transcription, ribonucleic acid is synthesized by complementary base pairing of ribonucleotides with deoxyribonucleotides to match a portion of one strand of DNA called a gene. During translation, specific transfer RNAs pick up specific amino acids, transfer those amino acids to the ribosomes, and insert them in the proper place according to the messenger RNA message. Bacterial and viral genomes act as pathogen-associated molecular patterns, or PAMPs, to stimulate innate immunity. And some antibacterial chemotherapeutic agents inhibit normal nucleic acid replication in bacteria. So those are our bullet points and our detailed learning objectives. Now, most of the stuff about DNA replication you've already had in your molecular genetics review exam, the take-home exam you did using a respondus test. So I'm going to cover on our Unit 1 exam DNA replication itself fairly lightly. You just need to understand the general process, a few of the enzymes involved. You won't have to know the details, but I am going to mention the details as we go through in case you had trouble following that on the take-home exam. So anyway, here's our detailed learning objectives for this SoftChalk lesson. Define genome. Describe the composition of the bacterial chromosome. Name the enzymes that enable bacteria to become circular, supercoiled, and unwind during DNA replication. Briefly describe the process of DNA replication. State the function of the following enzymes in bacterial DNA replication: DNA polymerase III, DNA polymerase II, DNA helicase, primase, DNA ligase. State the function of DNA. In terms of protein synthesis, briefly describe the process of transcription and of translation. Briefly state how the following antibacterial chemotherapeutic agents affect bacteria: the fluoroquinolones and trimethoprim sulfamethoxazole. So those are our bullet points and our detailed learning objectives you need to know for the exam. Let's get into the chromosome. So first of all, the term genome refers to the sum of an organism's genetic material. And as we learned when we talked about prokaryotic cells, the bacterial genome is composed of a single molecule of chromosomal deoxyribonucleic acid, or DNA. That chromosome is located in a region of the cytoplasm that we can see when viewed through an electron microscope, and that region is called the nucleoid. So here we see in figure 1 where the chromosome has already duplicated itself and separated prior to cell division. And this wad of DNA here that we can see, that area is called the nucleoid. Keep in mind that unlike the eukaryotic nucleus, the bacterial nucleoid has no nuclear membrane, and we see no nucleoli. So during DNA replication, each strand of the replicating DNA attaches to proteins at what will become the cell division plane, in an area where a division organelle called a divisome is made that we mentioned previously under the cytoplasmic membrane. And there are proteins called PAR proteins that function to separate the bacterial chromosomes to opposite poles during the cell division. These PAR proteins bind to the origin of replication of the DNA, and they physically pull and push the chromosomes apart, functioning similar to the mitotic apparatus that we see in eukaryotic cells. And this is a diagram we looked at under the membrane showing you the divisome and the two chromosomal molecules that have separated. And keep in mind that since bacteria are haploid, they have only one chromosome, or they are 1N. They only reproduce asexually, so they have there is no meiosis in bacteria. So the bacterial chromosome is one very long single molecule of double-stranded helical supercoiled DNA. And in most bacteria, the two ends of the double-stranded DNA covalently bond together, and they form both a physical and a genetic circle. So that's why we said back under prokaryotic cells that most bacteria have circular DNA, where the two ends of the DNA molecule bond together. Now, typically a chromosome in a bacterium is about 1,000 micrometers long and may have as many as 3,500 genes. If we take E. coli that we looked at in lab 1, at most that's only about 2 to 3 micrometers long. So it's a little box about 2 to 3 micrometers long, but that contains a chromosome approximately 1,400 micrometers long. So for that large molecule of DNA to fit into the bacterium, histone-like proteins bind to the DNA first. Remember, bacteria don't have the histones that eukaryotic cells, but they have similar proteins called histone-like proteins. And these histone-like proteins segregate the DNA into about 50 chromosomal loops or domains to make it a little more compact. And here's where the DNA topoisomerase enzymes come in. These are a group of enzymes. Again, they're called DNA topoisomerases. And they supercoil each one of those 50 or so chromosomal domains around itself to form a compacted mass of DNA about 0.2 of a micrometer in diameter. You can picture the coiling of those chromosomal domains like taking a rubber band and just wadding it up into a compact mass. And as the bacteria actively grow, in fact, there are projections of the nucleoid that extend into the cytoplasm, and that's thought to be where DNA is being transcribed into messenger RNA. And these DNA topoisomerases both insert and remove supercoils. To supercoil it, they cause it to compact it or wad up into a supercoil, but then they have to unwind in order for DNA to replicate. So these DNA topoisomerase enzymes are essential for the unwinding, replication, and rewinding of the circular supercoiled DNA. That tightly compacted circular mass has to unwind in order to replicate. Also, it has to unwind for transcription to occur. And then once that's done, it has to rewind to fit. So in order for that very long molecule of DNA to fit inside the bacterium, the DNA has to become supercoiled. And there's an enzyme called DNA gyrase, for example, that catalyzes this negative supercoiling of the circular DNA found in bacteria. But that supercoiled DNA must be uncoiled and relaxed in order for DNA polymerase to bind to the DNA for DNA replication, and in order for RNA polymerase to bind to the DNA for transcription. And so, for example, topoisomerase 4 is a topoisomerase that's often involved in the relaxation of the supercoiled DNA. Now let's get into DNA replication. Now again, my students taking this currently, you have limited objectives on that because we covered DNA replication, transcription, and translation in some detail on your review of molecular genetics exam from your prerequisite biology course that I provided you with SoftChalk lessons for. But in case you had trouble following that, because it is fairly complex, I do want to go through the process of DNA replication, although you won't have to know a lot of detail about that on our Unit 1 exam, just what the objectives ask of you. So let's start in a very general sense. DNA is replicated by uncoiling of the double-stranded DNA helix. As it uncoils, the two parent strands separate by breaking the hydrogen bonds between the two complementary strands. And then there's synthesis of new strands by complementary base pairing. And that's, of course, where adenine can form hydrogen bonds with thymine, AT or AT, and guanine can form hydrogen bonds with cytosine, GC. Keep in mind, in RNA, uracil replaces thymine. So first thing we need to realize then in this process of separating the two strands and making complementary copies is that DNA replication is bidirectional from a place called the origin of replication. So the origin of replication, which we see in figure 4 here, is where DNA replication begins in both directions around that long circular molecule. So to begin that, unwinding enzymes called DNA helicases cause short segments of the two parent DNA strands to unwind and separate. And that forms an origin of replication, forming two Y-shaped replication forks. So DNA polymerase or DNA helicase, shown in green here, started here, and it's unwinding the double-stranded helix in this direction at the beginning of the replication fork here, while another DNA helicase bound here and is unwinding the DNA in this direction. And that will continue all the way around the chromosome until it meets. So that's what the DNA helicase does. It unwinds short pieces of DNA, of the DNA helix, so that complementary strands can mate. So it does that by breaking the hydrogen bond. So notice this adenine here is no longer paired with this thymine. This thymine is no longer paired with that adenine. And that's where the new nucleotides will come in and line up by complementary base pairing. And figure 5 again shows you the bidirectional nature of that, where we see the two replication forks, where one helicase is unwinding the DNA in this direction, and the other helicase is unwinding the DNA in this direction. And in these unwinding areas, the replication fork, that's where the actual DNA replication is occurring, where the DNA nucleotides are lining up by complementary base pairing. And as the two strands unwind, there's some single-stranded binding proteins that bind to the unwound area so that the two strands don't rejoin. So an adenine doesn't repair with a thymine after the hydrogen bond was broken, for example. Now this does create problems because remember, DNA is helical. It's like a twisted ladder. And so as you unwind that double-stranded helix, that generates what are called positive supercoils ahead of the replication fork. There's a lot of stress from that helical molecule unwinding. So here's where topoisomerases come in. They put breaks in the DNA and then rejoin them to form negative supercoils to relieve the stress of that helical molecule unwinding. Now, as the two strands continue to unwind and separate in both directions around the entire DNA molecule, new complementary strands are produced by the hydrogen bonding of free DNA nucleotides with those of each parent strand. And as those new nucleotides line up by complementary base pairing or hydrogen bonding, then enzymes called DNA polymerase will join the nucleotides together, forming what's called a phosphodiester bond, or the phosphate of one nucleotide, the new nucleotide being inserted, will form a bond with the hydroxyl or OH group of a nucleotide already in the strand. Now, actually, the nucleotides that are lining up, the new nucleotides lining up by complementary base pairing, are actually deoxyribonucleotide triphosphates. Here we see a guanine deoxyribonucleotide triphosphate. It has three phosphates attached to it when it comes in. Now, when the bond forms, two of the phosphates are cleaved off, and they provide energy for forming this phosphodiester bond. So as the two phosphates are cleaved off, that allows the phosphate on this deoxyribonucleotide to bond to the hydroxyl group, not shown here, on the nucleotide already in the strand. And then eventually this phosphate will eventually bind to this nucleotide, and as the two phosphates are cleaved off, they'll provide the energy. And here we see the complementary base pairing. So you see that an adenine has hydrogen bound with a thymine, and then the phosphate on the deoxyribose is attached to the hydroxy of the previous one in the chain off here. Then a cytosine has hydrogen bound with a guanine. The phosphate of that deoxyribonucleotide has bound to the hydroxyl group here. A thymine is paired with an adenine. Phosphate binds to the hydroxyl group. Guanine pairs with cytosine, cytosine with guanine, adenine with thymine, etc. So that's the general idea behind DNA replication. DNA helicases unwind the DNA, and as the two strands separate, new deoxyribonucleotides come in and line up by complementary base pairing, and then DNA polymerase joins the phosphate group of the new nucleotide with a hydroxyl group of a nucleotide already in the strand. But as nature is, it's often more complex than that. And that's because of the nature of the enzyme DNA polymerase. DNA polymerase enzymes can only join the phosphate group attached to the 5 carbon of a new nucleotide to the hydroxyl or OH group on the 3' carbon of a nucleotide already in the chain. And what that means, and what we'll be seeing as we go through here then, is that DNA can only be synthesized in what's called a 5' to 3' direction. So it's being made in a 5' to 3' direction as it copies a parent strand that runs in the opposite direction, 3' to 5'. Now, at 5', 3' is not as complex as it sounds there. We need to know a couple of definitions. So keep in mind that each DNA strand has 2 ends. The 5' end of the DNA molecule is the one where there's a terminal phosphate group attached to the 5' carbon of the deoxyribose. If we look at figure 8 here, we see the left-hand strand. This is the 5' end because the last thing sticking out is a phosphate attached to the number 5 carbon of the deoxyribose. And the carbons are numbered clockwise here, so this would be the number 1 carbon, number 2 carbon, number 3 carbon, number 4 carbon, number 5 carbon. So the phosphate's attached to the 5 carbon, and that's called the 5' end. Now notice that the other end of the DNA strand, the last thing sticking out is a hydroxyl or OH group, and that's coming off the 3' carbon, 1, 2, 3. So that's the 3' end. So at one end, there'll be a phosphate sticking out, that's the 5' end. At the other end, there'll be a hydroxyl sticking out, that's the 3' end. but the two strands are anti-parallel in that they run in opposite directions because DNA can only be synthesized in a 5' to 3' direction. So notice the left-hand strand is running 5' to 3', phosphate here, OH down here, whereas the other strand is running 3' to 5'. There's an OH at the top here on the 3' carbon and a phosphate at the bottom coming off the 5' carbon. So we call that anti-parallel, where the two strands run in opposite directions. So that's the two strands running in opposite directions, anti-parallel. Now one of the two parent strands, the one that runs 3' to 5', because remember DNA polymerase can only copy a strand running 3' to 5'. It's making a strand running 5' to 3', the opposite. So the one that runs 3' to 5' is called the leading strand because this can be copied directly along the entire length. We'll show you what that looks like in just a second in figure 9. The other parent strand, the one that runs 5' to 3', is called the lagging strand because that has to be copied in short fragments, short discontinuous fragments called Okazaki fragments, where around 100 to 1,000 nucleotides are inserted as the DNA unwinds. Then as it unwinds some more, another 100 to 1,000 nucleotides are inserted, etc. So we get an idea of that in figure 9. Now this is our DNA helicase that is unwinding the DNA towards the left here. It started out down here at the beginning of the replication fork and worked its way down, separating the two parent strands. And it continues to unwind or unzip the DNA as it goes around the molecule. Now, as it unwinds, some DNA binding proteins attach, and that prevents these unpaired nucleotides, say maybe an adenine here, from repairing with a thymine down here. So that stops these unpaired bases from repairing with the one on the parent strand. Now, the helicase began here, so the replication fork, if we follow this strand, knows that this strand is running, this top parent strand is running 3' to 5', meaning the last base here has an OH coming off of the number 3 carbon, and the last base here has a phosphate coming off the number 5 carbon. So DNA is copied 3' to 5'. It's copying a strand, and it's made in a 5' to 3' direction. So the top strand, as it adds nucleotides, just keeps getting longer and longer and longer as it unwinds, and that's the leading strand. This is can be synthesized continuously in a 5' to 3' direction as it unwinds. However, the bottom strand, the bottom parent strand, is running 3' to 5'. So instead of a phosphate coming off like the top strand, there's an OH coming off here, and this is the one that has the phosphate on the end. And so this is going to be made in a 5', it has to be made in a 5' to 3' direction, but remember what's happening here is that that helicase unwound the DNA a little ways, let's say from here to here, but this is the strand that has to be made in this direction. So all it can do is copy that little short segment that was unwound. Then the helicase moves down a little further, unwinds some more DNA, but since this strand has to be made in this direction, again, it makes sure it copies what was unpaired by the helicase, then it unwinds it some more, copies another segment, unwinds some more, copies another segment, unwinds some more, copies another segment. So those are the lagging strands because it can only be synthesized in a 5' to 3' direction, copying a 3' to 5' strand. Then it has to be made in short segments as the DNA progressively unwinds. And eventually these fragments, or Okazaki fragments, on the lagging strand have to be joined together by DNase ligase to form a continuous strand. So one strand can be made continuously all the way around the molecule because that's the direction of unwinding, but the other one, because of the direction of unwinding, has to be made in short pieces. Every time it unwinds, another short segment has to be copied. And then they have to be joined together by DNA ligase. And then it gets more complex. DNA polymerase cannot begin a new DNA change from scratch. DNA polymerase can only attach new nucleotides onto a 3' hydroxyl group of a nucleotide already in the strand. But at the beginning of the replication fork, there is no DNA nucleotide in the strand at that point. So what has to happen here in both the leading and each of the DNA fragments of the lagging strand is that an RNA polymerase, an enzyme that makes RNA, called primase, is required. Now, primase can join RNA nucleotides together without requiring a pre-existing nucleotide in the DNA strand like DNA polymerase requires. So the first thing that happens at the beginning of the replication fork is that several complementary RNA nucleotides are lined up opposite DNA nucleotides by primase. And that forms what's called an RNA primer. And we see that beginning in figure 10. So notice the DNA replicates here. Remember, DNA can only be made in a 5' to 3' direction. So this strand is being made in this direction up towards the beginning of the replication fork as the DNA unwinds in the opposite direction. So it would start right here. And as the DNA helicase unwound it away, what the RNA polymerase or primase does is it complementary base pairs RNA nucleotides, ribonucleotides indicated by R for ribose rather than DR for deoxyribose. So an RNA nucleotide containing guanine would hydrogen bond with cytosine, and it can be inserted without having to bind to anything. Actually, this would be the first one. So cytosine would hydrogen bond with guanine, but again, this doesn't have to attach to anything with RNA primase. And then would copy this one, guanine cytosine, and may do a short sequence of them. Well, I'm only going to show you 2 in the diagram. So it starts out with primase, the RNA polymerase, initially winding up RNA nucleotides indicated by R with the DNA nucleotides indicated by D and I by complementary base pairing. And as each new one's added, the phosphate of the new one attaches to the OH of the previous one. But the first one doesn't have to attach to anything. Now, the same thing's happening at the beginning of the replication fork and the strand being made in the other direction. This is being copied in this direction, 5' to 3'. So again, an RNA nucleotide with adenine would hydrogen bond with thymine. Then uracil on the RNA nucleotide would bind to adenine, and then the phosphate would attach to the OH of the previous one. The first one doesn't have to attach to anything. So now the copying process has begun with a short RNA primer, a short sequence of RNA nucleotides complementary to the DNA nucleotides. So that's the primase role, to insert some RNA nucleotides complementary to DNA nucleotides at the beginning of the replication fork. And the same thing would be happening in this direction as the other helicase is going this direction towards the top. A RNA primer would be inserted. Then after that occurs, DNA polymerase 3 replaces the primase and is able to add DNA nucleotides to the RNA primer. So if we look at figure 11 here, so again, here's the strand that's being made in this direction from bottom to top, 5' to 3'. It's being synthesized, and here's the one that's being made complementary to the other strand running towards the bottom. So here's the 2 RNA nucleotide primers that we showed you that were inserted. But once a few of these are lined up, now DNA polymerase 3 can start inserting deoxyribonucleotides by complementary base pairing, and then hooking the phosphate of those up with the OH on the RNA primer. Because remember, DNA polymerase can only insert when the phosphate can attach to an existing nucleotide, be it DNA or RNA. So now DNA nucleotides begin to line up by complementary base pairing, attached to the RNA primer, and the same thing's happening here. Here's the beginning of our RNA primer that primase put in. So now DNA polymerase III starts lining up deoxyribonucleotides by complementary base pairing, and each one of those hooks to the OH of the previous one. Then another enzyme called DNA polymerase II digests away the RNA primer and replaces those RNA nucleotides with the proper DNA nucleotides. And we see that in the next illustration, figure 12. So here's where the two RNA nucleotides were located on this strand, and here's where the two RNA nucleotides were located on this strand. DNA polymerase II will remove the RNA nucleotides and replace them with deoxyribonucleotides, guanine pairing with cytosine, cytosine pairing with guanine, etc. So now the strand is all DNA nucleotides in both directions. And then the DNA fragments that are produced on the lagging strand have to be hooked together because remember, it's just made in short pieces, and they're hooked up by an enzyme called DNA ligase. So it's a pretty complicated process, and yet 1,000 micrometer long piece of tightly packed supercoiled DNA can make an exact copy of itself in about 10 minutes time in bacteria under optimum conditions. And it's inserting nucleotides at the rate of about 1,000 nucleotides a second. So it is very efficient. And if we go back up to a previous figure here, this kind of shows how one strand, again, the leading strand, the one that's being synthesized in a 5' to 3' direction, copying a strand running the opposite direction, 3' to 5'. This one, once the RNA primer is made, can then just be made continuously. But as the segments of the DNA unwind, remember the helicase started down here and is moving towards the left. This, as it unwinds, this has to be made in short segments. Then this has to be made in short segments as it unwinds some more. So it starts out by putting in RNA nucleotides for each segment using the RNA polymerase or primase. Then DNA polymerase starts adding DNA nucleotides. So the red are the RNA primers, the blue are the deoxyribonucleases. And so these fragments are made in that manner. Then eventually DNA polymerase II digests the RNA nucleotides and replaces them with complementary DNA nucleotides. So once it gets a certain point down here, eventually the RNA nucleotides are replaced with DNA nucleotides by DNA polymerase II. And then DNA ligase hooks all the fragments together eventually. And there's a couple nice YouTube movies here that indicate the process of DNA replication if you want to look at those, and a little self-check you can do. But again, a lot of that detail you won't have to know on your Unit 1 exam. Just do what the objectives say at this point. That brings us to the function of the bacterial chromosome. So, of course, we always say that the chromosome is the genetic material of the bacterium. And what we mean by genetic material is that genes located along the DNA are transcribed into RNA molecules. Transfer RNA molecules, ribosomal RNA molecules, and messenger RNA molecules. And most of the genes are transcribed into messenger RNA. And then the messenger RNA is translated into proteins at the ribosomes. So RNA copying the DNA is transcription, to copy or transcribe. And then ribosomes translate that message into proteins. Translation occurs at the ribosomes, where the messenger RNA is translated into polypeptide or protein. So again, we went through this in quite a bit of detail on your molecular genetics review exam. We had quite a few questions on that because we do apply that quite a bit. So let's just do a quick review of what transcription and translation is without getting into a lot of detail. So during transcription, ribonucleic acid, RNA, is synthesized by complementary base pairing of ribonucleotides with deoxyribonucleotides to match a portion of one strand of DNA called a gene. So remember, RNA is composed of the sugar ribose, one of 4 nitrogenous bases, adenine, guanine, cytosine, or uracil. Remember, uracil replaces thymine in RNA, and then, of course, a phosphate group. And deoxyribonucleotides are composed of deoxyribose as a sugar rather than ribose, and the 4 nitrogenous bases are adenine, guanine, cytosine, thymine, and then a phosphate group. So that's transcription, transcribing genes in the DNA into complementary RNA, such as messenger RNA. And there's a nice little Howard Hughes Medical Institute movie of transcription if you want to watch that. Now, during translation, specific transfer RNA molecules pick up specific amino acids, and they transfer those amino acids to the ribosome, hence transfer RNA, and they insert the right amino acids in the proper place in the growing protein chain by reading the messenger RNA message. And that's done again by complementary base pairing, where an anticodon, those are 3 ribonucleotide bases coming off the bottom of the transfer RNA molecule, will be complementary to 3 messenger RNA bases on the messenger RNA called a codon. So the anticodon portion of the transfer RNA binds by complementary base pairing with codons along the messenger RNA. And remember, codons are 3 consecutive messenger RNA nucleotide bases that code for a specific amino acid. We had all those terms on our molecular genetics review exam. Now, it turns out in bacteria, transcription and translation are coupled together. They're separate in eukaryotic cells because, remember, transcription, copying DNA into RNA, occurs in the nucleus, but then the RNA goes out nuclear pores to attach to the ribosomes. So the transcription and translation occur in 2 different locations. In bacteria, they're coupled together. RNA polymerase, the enzyme that's going to transcribe DNA into complementary RNA, binds to the 30S ribosomal subunit in the prokaryotic ribosome to form a transcription and translation machine, which they now call an expressome. So as the DNA is unwinding and being transcribed into complementary messenger RNA by RNA polymerase, that messenger RNA that's being made is fed into the translational center of the ribosome, where it's being translated into a polypeptide or protein. And getting back to our function then, when we say DNA is the genetic material, what we're really saying here is that DNA ultimately determines what proteins and what enzymes an organism can synthesize, because the order of nucleotide bases along the DNA determines the order of ribonucleotide bases along the messenger RNA, and the messenger RNAs determine what amino acids are inserted and in what order, and that determines the shape and function of the protein or enzyme. So DNA ultimately determines the primary structure of a protein or enzyme, and because certain amino acids can pair with other amino acids in the chain, that determines the shape and the function of the enzyme. And of course, the enzymes then determine what chemical reactions that cell can carry out, and those chemical reactions determine the trait. So DNA ultimately determines the proteins and enzymes made, which then determines the chemical reactions that can be carried out, which then helps determine traits. And there's some nice animations on YouTube showing translation if you want to look at those, and a little self-check you can do. I want to talk briefly about the bacterial epigenome. This is a layer of genetics higher than the genome being the chromosome. The epigenome refers to a variety of chemical compounds that can modify the genome. And there's various ways this can be done. One way a genome, and remember a genome is a sum of an organism's genetic material, one way the genome can be modified is by adding a methyl or CH3 group to nucleotide bases at specific locations along that DNA molecule. Now, when that methyl or CH3 group is added, we call that methylation, adding a CH3 group. that methylation can in turn either repress or activate transcription of specific genes. So where this methylation occurs, where adding this methyl group occurs, can determine whether a gene is activated and transcribed into messenger RNA or whether that gene is repressed and not transcribed. Now, another way the genome can be modified besides adding chemical groups like methyl groups is by attaching chemical groups to the histone-like proteins in eukaryotic cells or the histone-like proteins in prokaryotic cells. And remember, the DNA winds around these histones and histone-like proteins, and the way it winds around determines how tightly bound it is or loosely bound it is. So modification of the histones or the histone-like proteins can also repress or activate transcription of specific genes. If the DNA is very tightly wound, then it can't be transcribed into RNA very easily, where if it's more loosely wound, then it can be transcribed. And this is an essential part of organisms being able to function. By basically turning various genes within the cell on or off by whether or not methyl groups are added or by how the histones are or histone-like proteins are modified, the epigenome can, in the case of humans, determine what types of cells carry out what functions. So remember, all cells in the human genome contain the same DNA. But different cells need to carry out different biochemical functions. So of course a muscle cell is going to be carrying out different functions than a liver cell. And to function as a liver cell, it needs different genes activated or different genes repressed. And it's the epigenome that allows that to happen. So the epigenome is actually used to determine what genes in a cell are being transcribed and are functional, being activated, and which are being repressed, and that in turn can determine the function of that cell. And we also know now that parts of the epigenome can actually be inherited. At one time it was thought that during meiosis that the epigenome was rebooted, all those chemicals were removed, but we now know that in response to the environment or in response to lifestyle, that things like smoking or exposure to pollutants or diet or infections can actually affect the epigenome, and sometimes that epigenome doesn't reboot but actually becomes inherited. Now it becomes inherited is important in many cell functions, so that a muscle cell pretty much always does what a muscle cell does, that part being inherited, but it can also adversely affect the genome, causing harmful effects, such as can occur with exposure to certain chemical pollutants or mutagens. Well, just like the human epigenome affects human genes, the bacterial epigenome affects bacterial genes. But the bacteria can actually affect our epigenome and modify the function of our genome. So bacteria may cause either DNA methylation or modification of our histone proteins, the bacteria leading to that, and that resulting modification of the human genome by the human epigenome by the bacterial genome can either activate various genes involved in immune defenses or maybe suppress immune genes. So it may be functioning to activate certain genes so that whereas we recognize the bacteria and we want to mount a strong immune response against it, but sometimes the bacteria may affect our epigenome and turn off immune response genes so we can't fight the bacteria as well. So there's many things in nature that can affect the epigenome, like we said, from lifestyle to environment. Anything from cigarette smoke to exposure to pollutants to diet to various infections. And there's a nice little article there that gives you some good examples of that if you want to read it, and a little YouTube video on the epigenome if you're curious. But we just are introducing the concept of it here. Now, it turns out that the bacterial chromosome can actually initiate our body defenses. So remember, as we learned previously under the cell wall SoftChalk lessons, To protect against infection, the body needs to be able to detect the presence of microorganisms that might be causing an infectious disease. So we do that initially during innate immunity by recognizing molecules unique to the microbe that human cells don't have. And again, we call these pathogen-associated molecular patterns, or PAMPs, molecules with a certain shape, molecular pattern found on the pathogen. But it turns out that both bacterial and viral genomes can initiate or activate innate immunity, because bacterial and viral genomes have a high frequency of what's called unmethylated cytosine guanine dinucleotide sequences. These are terms that microbial geneticists come up with, god-awful long terms, but they make perfect sense. They're just god-awful long. So this says, what this tells you is that cytosine guanine dinucleotide, that would be two nucleotides joined together, which would be cytosine followed by guanine. But in bacterial and viral genomes, that cytosine doesn't have a methyl group attached. Now, mammalian DNA, our DNA, has a lower frequency of cytosine guanine dinucleotides. So we have fewer C's followed by G's along the DNA, and most of our cytosines are methylated. So if we look at figure 13, this is a methylated cytosine, where there's a methyl group right here coming off. Now, in bacteria and viruses, that cytosine doesn't have a methyl group here. So those are unmethylated cytosines. And our pattern recognition receptors can actually tell that difference. We have pattern recognition receptors that can determine whether that cytosine has a methyl group or not on it. And since there are more C's followed by G's, and the C's are unmethylated in bacteria and viruses, then those can easily be seen by the pattern recognition receptors. And so once those unmethylated cytosine guanine dinucleotide sequences are recognized by our pattern recognition receptors on our defense cells, that's going to trigger innate immune defenses like inflammation, fever, and phagocytosis, just like cell wall PAMPs do. And there's a little self-check you can do on that. Now, there's also some antimicrobial agents we use that inhibit nucleic acid replication in bacteria. And we're going to talk about these more in Unit 2 when we get to control of bacteria, but to introduce that concept, the two groups that we're going to talk about primarily are a group of antibiotics called fluoroquinolones. These are antibiotics that have an oxacin or a floxacin in their name. For example, one common brand of antibiotic is Cipro, which is actually ciprofloxacin. Well, all of these antibiotics work because they inhibit one or more of the topoisomerase enzymes that bacteria need to supercoil and uncoil their DNA during DNA replication and during transcription. So by interfering with those topoisomerase enzymes, they block nucleic acid synthesis. And what they do then is they actually alter the topoisomerase enzyme. For example, if we look at a normal enzyme reaction in this animation, this would be the active site for the enzyme that would bind to a substrate. So in that case, it might be a topoisomerase enzyme that's either supposed to coil the DNA or uncoil the DNA. But these antibiotics like fluoroquinolones work because they have a shape that fits part of that topoisomerase enzyme. They have a shape that fits another part of the enzyme at a different location. So if the drug is not bound to the enzyme, then the enzyme functions normally. It takes the substrate, does whatever it's supposed to do to that substrate. The enzyme functions. What happens when the antibiotic is present is that the antibiotic, in this case like a fluoroquinolone, binds to a shape, fits a shape on the topoisomerase enzyme. And when the antibiotic binds to the enzyme, that changes the shape of the enzyme so it no longer functions as long as the drug is bound. And now the topoisomerase can't carry out its normal activity of either supercoiling or relaxing the DNA during DNA replication and during transcription. And then our second group that we'll talk more about later on in Unit 2 is a drug is called cotrimoxazole. Bactrim and Septra are two brand names of that. And this is a combination of two synthetic drugs, sulfamethoxazole and trimethoprim. And these two antimicrobial agents alter two enzymes in the bacterial pathway required to synthesize a cofactor needed for DNA replication. That cofactor is called tetrahydrofolic acid. So for bacteria to make most of the nucleotide bases, thymine, guanine, uracil, adenine, they require tetrahydrofolic acid as a cofactor. If that cofactor is not made because the enzymes involved in making that cofactor are blocked by the drug, then the bacteria can't synthesize the building blocks it needs for DNA replication or RNA replication. And we'll see more about how that works later on in Unit 2. But there are a few agents that do actually inhibit nucleic acid replication in bacteria. Not too many because, again, bacterial DNA is not that different from our DNA. And finally, at the very end, we have a self-quiz over the whole chromosome and nucleoid SoftChalk lecture if you want to look at that.